So, ladies and gentlemen, thank you very much for joining us. I'm Dr. Joel Mortensen. I'm the director of the Diagnostic Infectious Diseases Testing Laboratory at Cincinnati Children's. I'm also the assistant director of microbiology at Doctors Data. I have some obligatory pieces to share with you. Yes, I'm at an academic institution. We do grants and research support. I'm on the Speakers Bureau for a couple of different companies. I'm on some consultant and advisory boards, and as I said, I'm the assistant director of Doctors Data. So today we wanted to talk a little bit about what are some questions regarding stool testing. And we get lots of calls from the field, and so I wanted to jump into some new things that we have that I think will answer some of those questions, and we'll see where this all goes. So first, what's the state of the art of microbiome analysis, PCR, culture, combination testing? How does that all fit together? And what's the role of PCR and culture in detection of true stool pathogens, classic pathogens, and antibiotic resistance testing? So let's start with the state of the art of gastrointestinal microbiome testing. And as you all know, there's a number of functions that are impacted by the microbiome. So there's a lot of connection between us and our gut flora. Not surprising. So there's a lot of host physiology that goes on, adaptive immunity, innate immunity, biosynthesis of neurotransmitters, steroids and hormones, vitamins, metabolism that we're interacting with our microbiome. In addition, there's a lot of evidence that diseases are related to aberrant processes in our microbiome. So allergies, heart disease, cancer, diabetes, autoimmune and inflammatory diseases, neurodevelopment, and lots of other things have been associated with microbiome. There's a lot of work going on to try to establish cause and effect. And in fact, at Cincinnati Children's, we're involved in microbiome research, as a number of institutions are. And we've done descriptive work, again, like others. And some of this now, 10 years ago and more, was revolutionary, because what they taught me was normal flora of our gastrointestinal tract is they lied to me. This is what normal children's microbiome at the phylum level, and remember phyla is the highest level below kingdom for classification. So the major constituents are Bacteroidetes, Firmicutes, a little bit of Proteobacterium, and then Actinobacterium. And this represents thousands of hours of work to get there. You can also drive this down further if you have an interest into some of the very specific genera or families or species that are involved. So Bacteroides, again, makes up the majority of our normal flora, but Faecalibacterium is an agent of interest, as is Bifidobacteria, Eubacterium, and you can see that Escherichia, the classic E. coli, the normal flora, makes up a very, very small percentage of normal flora. So again, work that we've done at Cincinnati Children's and others have done has looked at fecal microbiome in recurrent C. diff disease. And so again, this is just a different way of looking at the microbiome that we got. This particular study, we sort of described them and displayed them a little bit differently. But this is the normal microbiome. And the radical difference becomes when you look at that microbiome in a patient with chronic C. diff disease. All of a sudden, the Proteobacteria, E. coli, Klebsiella, Citrobacter, Morganella, Providencia, all of those now are taking over the gastrointestinal tract, and Firmicutes and Bacteroidetes is now a minority. That's a problem. And in fact, there's no question that patients with recurrent C. diff disease, be they pediatric or adult patients, have decreased phylogenetic richness. They don't have the representation, the diversity, and the depth that our normal patients have. And it is the Bacteroidetes and the Firmicutes that are significantly reduced in recurrent C. diff. Now, if you compare that to patients with a single episode of C. diff disease, they don't have this massive change in their microbiome. So we spend a lot of time describing. We follow patients after transplants and do a lot of interesting work. But I think the key, and what I wanted to really spend some time with today, is bridging the gap between research and the clinical world in which you all live. So my goal was to work on getting a standardized and routine tool to profile the microbiome and to link this to clinically actionable items. So we want to identify some profiles to aid in the diagnosis as well as selection of treatment. And I think that we can identify profiles as major risk factors for the development of a variety of diseases. So doctors' data, in that effort, we just started using a dysbiosis test, which was originally developed by Genetic Analysis, a company out of Norway. And this dysbiosis test is intended to be used as a gut microbiome DNA analysis tool. It allows us to characterize and profile. Highly reproducible test. It is CE marked, has an ISO. The CE mark, by the way, is just the European equivalent of the FDA clearance. And we're not going to spend a lot of time doing really fun, geeked out science that I love to tell people about and no one really wants to hear about. But essentially, we take a stool sample, we extract the DNA, we use this set of primers provided, do a classic set of PCR, and are detecting in the Luminex system. So it uses 54 sets of primers for specific groups of bacteria and specific species of bacteria that were selected to differentiate patients with normobiotic versus dysbiotic microbiome. So we're not simply using a variety of primers to describe what's there, but the original design and intent was to separate patient populations, normobiotic to dysbiotic, and we'll talk a little bit about that. So, of the key characteristics that I started out listing of things we wanted to accomplish, let's start in the laboratory and give you some idea of the basis from the laboratory's perspective of the test. So, this graph represents 6 patient samples run in triplicate for 3 different lots of kits and repeated 3 times. So what you see is fuzzy colored circles. Mine's a little better focused up here. But anyway, these are our 6 patients. They all are different, but the clustering shows you that each of those individual runs and lots and kits and repeats all cluster together. Marvelously reproducible test. And in fact, if you get into the technical verification, we looked at repeatability, which is the measure within an individual run, and reproducibility, which is variability between runs, and looked at the coefficient of variability. And so within runs, it has a 2% coefficient of variability, which is a very small variability. And then between runs, it's a little bit higher, but still marvelously consistent in the testing. So the lab is happy, but that's not clinical. So what kind of clinical validation is there? So the dysbiosis test, again, is based on differentiating a normobiotic group from a dysbiotic group. And this is based on at least 500 samples to date in separating in the ability to separate those populations. And the study that I show here, which came out in 2015, took the clinical definitions of IBD, IBS, and healthy. It included a significant number of patients in each of those groups, and then asked the question, can this test differentiate dysbiotic from normobiotic? So one of the analogies that I've used is in diabetes, we can do a blood glucose level. We can take an individual test result and get an individual number. But it doesn't really give us the answer that we're looking for. What we're looking for is something more like the A1C, which allows us to give more of a trend, clustering in the groups, separating populations that may be at risk for developing more disease, or those populations that have a trend that's not going to develop those same kinds of problems. So the test, differentiating dysbiotic and normobiotic, you can see separates IBD, IBS, and healthy patients very, very nicely. Now you might also say, well, that's not 100%. No, it's not. None of the tests are 100%. A1C is not 100%. but it does a very clean differentiation of the dysbiotic and the normobiotic population. So that's the original clinical validation for the assay. So beyond that, there's a number of peer-reviewed publications. There's more than 20 that I've read. There are more beyond that. There are clinical data now for 5,000 patients, and there are over 50 clinical trials that have used this particular test to support their individual test protocols. And what I wanted to do is just share a couple of those articles with you to give you a sense of what's going on in the research world applying this dysbiosis test. So this first study asks the question, can fecal microbiome profiling be used in pediatric patients with IBD compared to children with gastrointestinal symptoms that is not IBD and compared to healthy children? So essentially, microbiome profiling. The design was 233 children and adolescents who had inflammatory bowel disease, and there was 110 with multiple subgroups that were clinically differentiated. Children with symptoms of gastroenteritis, 50 and 75 healthy children. The treatment was determined individually, and the microbiome profile was used by the dysbiosis test, and then the patients are followed over time. So key findings. Patients with gastrointestinal symptoms had reduced abundance of 51 of 54 key bacterial groups and species compared to healthy controls. So these two groups over here, compared to the healthy group over here, were significantly different. And Firmicutes was significantly lower in IBD versus the symptomatic. So the difference between these two groups right here, Firmicutes was significantly lower in IBD, specifically Eubacterium and Bifidobacterium. In addition, the need for surgery and the lack of mucosal healing was associated with a higher abundance of Proteobacterium, again our old style normal flora, E. coli, Klebsiella, and Citrobacters. So this group that required surgery into their course was significantly different than no surgery. Proteobacterium were higher, and Faecalibacterium prausnitzii was lower. So the conclusion of this study was that prior to treatment, bacterial composition of the microbiome of patients with IBD was very different from healthy children. And the degree of disturbance of the diversity and abundance of the bacterial microbiome in children with IBD correlated with the disease course and the need for later therapy. Patients with higher numbers of proteobacteria were more likely to need aggressive treatment and surgery. So a second study, looking at the clinical course, because the observation of the authors were that for them, the clinical course of ulcerative colitis varied substantially between individuals and couldn't be reliably predicted. So they designed a study where they had stool samples obtained from 18 newly diagnosed patients with ulcerative colitis prior to any therapy being introduced. And then they again were followed over time, looking to see whether or not the disease progressed mild or moderate to severe. And they followed them for a 3-month period. So key findings from this study were that the dysbiosis test separated those that would be classified as mild to moderate, and that's shown here in this multivariate analysis. So although there's a teeny bit of overlap, one outlier, from the multivariate analysis, the two populations were separated clearly. No single bacteria, though, defined these groups. So you can't simply say it's all E. coli's fault, or Klebsiella is the best marker, or the newest study says one single organism. However, higher abundance of Proteobacterium, strep species, were associated with moderate to severe disease. So again, too much Proteobacterium is problematic, And higher levels of Ruminococcus and Akkermansiae were seen in the mild disease, more closely mirroring a normal population. So in conclusion for this study, patients with newly diagnosed ulcerative colitis, the fecal microbiome diversity was linked, not cause and effect, but was linked to the disease course, and the determination of key bacteria, the abundance of key bacteria, may be an important marker to further differentiate the course of the disease development, which was the goal of that study, was an effort to say, how do we think this is going to progress? The last study that I wanted to just share with you briefly was about treatment. There's a variety of treatments that are used and tried, but one of them is anti-tumor necrosis factor therapy for patients with ulcerative colitis. Not all patients respond well, and the question from this study was, is there a way that the microbiome can help the authors separate those that are going to respond to anti-tumor necrosis factor and those that are not going to respond? So the design of this study was multifactorial. So serum and biopsies were obtained from 56 patients with ulcerative colitis, again, before starting therapy. Stool samples for microbiome analysis were only available, unfortunately, for 7 patients, but we were able to track them over time as well. The study included looking at antimicrobial peptides and cytokines. They were measured both in serum and in tissue. The microbiome profile, again, was determined using the dysbiosis test that I've been talking about. So the patients then went on to be treated with anti-tumor necrosis factor, and the treatment response was determined over time. And in this case, the Mayo scoring system was used and required an index shift of more than 3 to be considered significant. So looking at the results from this study, patients that ultimately responded to anti-tumor necrosis therapy, responders again versus non-responders, were distinctly separated in this multivariate analysis by the gut microbiome profile that was obtained before the patient started therapy. So there was an ability to predict. And again, the abundance of Faecalibacterium prausnitzii tended to be higher in patients that were responders, a bit closer to normal biome than in the non-responders. Their conclusions were that populations that may respond to biological treatment of ulcerative colitis can be separated using the dysbiosis index. This test also allows specific groups of bacteria to be further looked at beyond just the overall dysbiosis score, and potentially can be used for helping us, those specific organisms, in predicting a response to at least anti-tumor necrosis factor. So the next big question that I wanted to touch on, that's related but different, is the role of PCR culture in detecting classic pathogens from stool. So traditional culture provides a significant amount of data through the use, in the case of doctors' data, MALDI-TOF for flora and pathogen identification. And since I'm old, I'm very traditionally based in the culture world, so I personally prefer cultures for a lot of different things. Louis and I used to work together, in case you wondered. Louis Pasteur. Fine. It's going to go like that today. Anyway, nucleic acid amplification, such as PCR, though, has added to our ability to detect individual pathogens with very, very high sensitivity and has revolutionized the ability of detecting conventional pathogens. The limit, of course, is that you can't find something you're not looking for, so PCR has to be designed very, very carefully. But what about PCR for pathogens? Well, the development in the last 10 years of a variety of FDA-cleared and highly validated platforms has really provided reliability for detecting stool pathogens across the board and has, in some cases, revolutionized our ability to detect things like Campylobacter, Salmonella, and Shigella. And standardization of these platforms is what has led to the high sensitivity and high reproducibility that we can use today on a variety of FDA-cleared platforms. They are a variety of very effective tests for pathogenic bacteria, for viruses, and parasites, and those menus continue to expand. So as I said, culture is still the gold standard. Isolated organisms also are required for standardized antimicrobial susceptibility testing. So if you only are using a PCR-based assay, you're not going to have an organism if you need to move into therapy. It also, conventional culture allows for things that we don't know that we're looking for. So we find unique organisms such as Archobacter species, unusual yeasts in a variety of stool specimens that if we were limited to PCR, we wouldn't have the ability to detect them. And the state of the art today is the use of the MALDI-TOF, which is mass spec time of flight. Again, I would be happy to spend time talking about what all that does, but very few of you would care. But it has revolutionized our ability to identify organisms because the present databases have probably 1,500 genera and species of bacteria and fungi, which again is significantly different than it was 10 years ago. The next question I wanted to touch on is I get fairly frequently, which is, can PCR detect antibiotic resistance? And so the cartoon that I had drawn for you today is a bucket, and it's full of stool. Now, originally I had a picture of a bucket full of stool, and then I was told, no, this is lunchtime. Oh. So now it's a drawing. But imagine a bucket full of stool. And floating around in that bucket are bits of DNA and RNA from a host of different viruses, bacteria, parasites, as well as antimicrobial resistance markers with lots of cool names like ESBL and CARBA and other things. The question is, can you do susceptibility testing by sampling or dipping into that bucket? And the answer is, unfortunately, no. Susceptibility testing of bacteria and yeast requires standardized methods of isolated organisms. If you simply go looking in stool without an associated organism, there's no way that it can be used to guide therapy. Because in fact, we've done studies on what we call the resistome of gastrointestinal tract, and you can find literally hundreds of resistance markers in normal stool of completely well patients due to previous exposure to antibiotics, the environmental exposure, and resistance mechanisms of non-pathogenic bacteria. For example, Leuconostoc and Pediococcus. And by the way, if any of you wonder why you constantly see new names of bacteria, microbiologists get together once a year. We change 10% of all bacteria's names just to confuse clinicians so that we have the secret handshake and you don't. That is a joke. Maybe. Anyway, viable Leuconostoc and Pediococcus are found in fermented foods. All Leuconostoc and Pediococcus are vancomycin resistant. So if you use a sensitive probe and go into stool of anyone who's ever eaten a fermented food, chances are there's a Pediococcus or Leuconostoc, and you'll get back the reading that the stool has vancomycin resistant organisms. Yes, and it's a good thing. It allows me to make sauerkraut and kimchi, and that's what you need. So simply fishing for resistance markers is not a helpful process. So let me go back. How is doctors' data involved now with bridging this diagnostic gap? Combining key elements to give us a standardized and easy-to-use tool to identify profiles and help develop a way to identify risk. And so this is the first page of the report that's available from doctors' data. And if you visit the doctor's data booth, there's additional information. There's some additional information at the tables. But this is the beginning of a revolution for us in integrating multiple modalities of testing to give you the best answer we possibly can. We've incorporated the microbiome test that I was talking about earlier. This is the GI 360 test. So this first graph summarizes the use of that test. So, for example, if you have less actinobacterium, it's drawn in from our normal biotic. The normal biotic is green. It's drawn in. If you have more proteobacterium than the normal biotic control group, then it would be drawn out, as in case here in the Bacteroidetes. So we've taken key phyla and groups compared to that normal population and says, how does this patient look in comparison? Now, that's just an overview. We can actually draw that down and split out individual groups of organisms if there's an interest or a need that you want to go beyond that dysbiosis summary. So as you can see here in the center, it's green. 0 is our normal biotic population. And as the patient varies more, +1, +2, +3, we go from yellow to red. Or as they get less than our normal biotic, -1, -2, and -3, it goes down. And that's a graphic representation of our individual patient for a whole host of organisms. So in this case, this happens to be the phyla Actinobacteria, and we can look at Actinomyceta thales, we can look at Bifidobacterium, we can go into Bacteroidetes, and on down from there, a variety of individual organisms. In fact, this represents all of the individual groups of organisms. And originally I was going to talk about each and every one of them individually. I was told I was not allowed to. But there's lots of information here that we can use if we're following individual patients or if we want to drill down into individual groups of organisms like Proteobacterium. In this new 360 test, we then also combined the microbiome data with multiplex PCR for key viruses, bacteria, and parasites. And again, it brings the very high sensitivity for clear, known pathogens. And finally, we have conventional culture used to supplement those other tests to allow for the isolation of infrequent or unusual bacteria and also to allow us to do antimicrobial susceptibility testing. So you can see here, organisms like Salmonella and Shigella would be picked up by the PCR, but here we can also do susceptibility testing if we need to do that. So what is the state of the art? My view is it's PCR used to determine the abundance and diversity of bacteria, the microbiome, specific PCR primer sets to detect known pathogenic bacteria, viruses, and parasites, combined with comprehensive methodologies. So, for example, we continue to do comprehensive parasitology, a medical technologist sitting at a microscope, because there aren't PCR primers for all known parasites. This allows us to pick up worms, unusual and rare parasites, and other variations on that would not be picked up by a PCR assay. We also then continue to use stool culture with MALDI-TOF for identification, as I said, for susceptibility testing of bacteria and yeast, and the identification of unusual organisms. Finally, we do offer a broad range of stool chemistries. I'm a microbiologist. Please don't ask me about chemistry. I have really no idea. Someone does. So, of course, the last question is, great, you've given me a test. What do I do with it? And Dr. Kalish will join you all tomorrow at lunch to talk about interpretation and applying GI test data in GI dysfunction. So thank you all for joining us.